Hello students, welcome to Zoology Classroom. In today's class, we are going to know about nervous tissue. Actually, in our syllabus, we have this structure of neuron, and many of your friends have taken seminar on this topic, and you are being studying about this structure of neuron since your high school. So, and many of the things related to this topic are known to you. As you know, I am not going to repeat all of them, but I would like to tell you some new things related to this topic. Okay, so in this structure of neuron, we know that it will have a soma, axon, and dendrites will be there. Okay. And in soma, again, cytoplasm will be there, nistal granules will be there, nucleus, and all those things will be there. Okay. And dendrites, two types of dendrites will be there. One type of dendrites, they originate from this soma, and at the end of this axon also telodendrites will arise. So, and some neurons, they will possess this myelin sheath around this axon. So all these things are known to you. So I am skipping this details. Now let us move on to next topic. This is very, very important. The thing that I am going to explain now is not in our syllabus, but for our understanding, it is very, very important to know that. So in our body, different types of tissues are there. Different types of tissues are there. Out of those tissues, nervous tissue is one. And nervous tissue is very, very important one. This nervous tissue is there in our body to know what is happening around us. That means outside our body, to understand the consequences, and to help the animal to survive in the outside world, to survive the in the environment. This nervous tissue sees that some coordination will be there in our body. So different types of organs, organ systems, tissues are there in our body. So all these tissues and organ systems, they need to be functioned in a coordinated way. Okay, all the organ systems have to function in a coordinated way. So all the organs in our body, they cannot function at their own will. Okay, so they have to be work in a coordinated way. So just like in a cricket game, if you want to win the match, all the 11 members of the same team have to play the game in a coordination manner. If there is no coordination in the fielding team, or if there is no coordination in between the two batsmen who are batting on the field, what will happen? If there is no coordination in between this batsmen, there is a chance of getting run out. Okay, and if there is no coordination between the fielding team, they may slip the catches or they may give excess runs like that. So if there is a coordination, the play will be good and they will win the match. So like that, in the same way, different organ systems in our body also, they have to function in a coordination manner. So there should be a coordination in the functioning of all the systems. So the coordination duty is assigned to this nervous tissue. And the coordination function will be done mainly by this brain, central nervous system. Okay, in central nervous system, no brain, spinal cord will be there. And in peripheral nervous system, 12 pairs of nerves coming from this brain, cephalic nerves, and 31 pairs of nerves coming from the spinal cord. So the spinal nerves, so cranial nerves and spinal nerves, these two, they will make the peripheral nervous system. And some special neurons will be there in this just beside this spinal cord. So separate special neurons and separate nerve ganglia will be there. They will form this autonomic nervous system. And this autonomic nervous system also uses the components of the central nervous system and peripheral nervous system also. So this is the structure of nervous tissue in our body. So how this nervous tissue is made up of? So this nervous tissue is made up of a structural unit called neuron. So all the neurons will be there, many neurons will be there, and aggregation of those all neurons present inside our body, it will finally give this type of structure, like the brain, spinal cord, cranial nerves, spinal nerves, and autonomic nervous system ganglia. So this is the structure of our nervous tissue in our body, and basic structural unit of our nervous system or nervous tissue is neuron. And we know the structure of neuron very well. So apart from this neuron, several other accessory neurons also will be there in the nervous system, in the nervous tissue. And these accessory neurons, or we may call them helping neurons, they are like astrocytes is one type of helping neurons or accessory neurons, microglial cells, and oligodendrocytes. And the myelin sheath, we know about this myelin sheath. Myelin sheath is also a type of cell actually. This myelin sheath is made up of a Schwann cell. Schwann cell is another type of accessory neuron or helping neuron. So microglial cells, oligodendrocytes, Schwann cells, and astrocytes. So these are the 4 different types of accessory neurons or helping neurons. So the main function of the nervous system will be performed by these neurons only. But these accessory neurons, they will help these neurons in performing their function. So these oligodendrocytes, microglial cells, astrocytes, and Schwann cells are helping the main neurons in performing or in discharging their duties. So like this, in the nervous system, not only the neurons, apart from these neurons, several other accessory nerve cells also will be there. They are microglial cells, astrocytes, myelin sheath, and oligodendrocytes. So they will be surrounding this original or main neurons in the nervous tissue, in the nervous system, and they will be helping this nervous system by supplying nutrients, by supplying nutrients or food materials required, oxygen required for these neurons, and Schwann cells like Schwann cells, they will provide insulation to the neuron so that the action potential will be propagated in a rapid manner. So like this, all these cells help these neurons in one or other way. And in the neuron structure also, depending on the structure, the neurons can be categorized into bipolar neuron, unipolar neuron, or pseudo unipolar neuron, and multipolar neuron. So we have been studying about the structure of multipolar neuron. Okay, we know about the structure, and remaining things are also the remaining 2 types of neurons are also will be having the same parts in their cells, but the only difference is that the type of the number of dendrites, the number of axons will be different in these 2 different type of cells. So except these changes in remaining aspects, all these 3 are same. So depending on how many axons are there, okay, depending on how many axons are there, the neurons are categorized into bipolar neurons, unipolar or pseudo-unipolar neurons, and multipolar neurons. Apart from this, neurons can be categorized into 3 other different types also, depending on in which direction they are sending signals. Based on this feature, neurons can be categorized into sensory neurons, motor neurons, and interneurons. Sensory neurons, motor neurons, and interneurons. Sensory neurons, they will send signals from body parts to brain. And motor neurons, they will send signals from brain to body parts. And some mixed neurons will be there. mixed neurons, they will send the signals in both directions. That means they will send signals from body parts to brain and from brain to body parts. So in both ways, in two ways they can send the signals. Such type of neurons are called as mixed neurons. And fourth type of neurons are called as interneurons. And these interneurons, they will be especially they will be present in this autonomic nervous system. They will be acting as a temporary decision makers. So interneurons are another type of neurons. They will be present in the this what autonomic nervous system. So these are the four types of neurons based on in which direction they are sending signals. And depending on whether the myelin sheaths are present or not, depending on that feature, neurons can be classified into two types. Like one is myelinated neurons. We are seeing in this second one, this is myelinated neuron. And another one is non-myelinated or unmyelinated neuron. And unmyelinated neurons will not be having any myelin sheaths. In this neuron, we can see 1, 2, 3, 4, 5, 6. Total six myelin sheaths are there. And each myelin sheath Okay, each myelin sheath is a separate Schwann cell. So totally 6 Schwann cells are there. Each Schwann cell is forming a myelin sheath around this neuron. And the first neuron, it is lacking these myelin sheaths or Schwann cells around its axon. So it is called as unmyelinated or non-myelinated neuron. Here, what is happening in these myelinated neurons, while the action potentials are propagated from this soma to this telodendrites region, the speed of the action potential propagation will be very great in this myelinated neuron. Whereas in this non-myelinated or unmyelinated neuron, speed of the propagation of action potential will be very slow. So in this action potential travels in a slow manner. In this myelinated neuron, action potential travels very fastly or swiftly. So fast, it will be traveling in a speedy manner. Okay, so this is the difference between these two types of neurons. So depending on whether myelin sheath is present or not, two types. One is myelinated, and the one is non-myelinated. And depending on how many axons or how many dendrites are there, based on this thing, three types. One is bipolar, second one is unipolar, and third one is multipolar. And depending on in which direction the neurons are sending signals, based on that feature, four types, we can say four types. One is sensory neurons, another one is motor neurons, third one is mixed neurons, and fourth one is interneurons. And coming to the structure of nervous tissue, we know that in this slide you can see this. This is the section of spinal cord. Okay, this is the section of spinal cord, and you know very well about this structure of the spinal cord, internal structure also. So in this thing, internally, this H-shaped blue gray matter will be there, and outside white matter will be there. So how this white matter and how this blue matters are being formed, and how this is nerve. Okay, the nerve coming from this spinal cord is called as spinal nerve. So how the spinal nerves are being formed. So let us know about this thing. So this is very important to understand the structure and arrangement of nervous tissue. And in the beginning, in this slide also, we know this neuron brain is there. So how the brain will be formed with the congregation of neurons. Let us know about this thing. So this is a single neuron. Okay, let us forget about this, whether it is a multipolar neuron, unipolar neuron, or bipolar neuron. So different types of neurons will be there. And coming to this structure of neuron, let us once again remind this thing what cell granules will be there in this cytoplasm or inside this soma. Okay, in the neuroplasm, Nissel granules will be there. This Nissel granules will be gray in color. So due to the Nissel granules only, the soma will appear gray in color. And due to this axons are surrounded by many of the neurons in our body. Many of the neurons in our body are surrounded by this myelin sheath. So in our nervous system, mostly 95% or more than 95% of neurons are of myelinated type, and only few number of very less number of neurons belong to unmyelinated type of neurons. So, and many of these neurons, as the axons of them are surrounded by myelin sheaths, and myelin sheath appears in white color. So remaining total body of the neuron, almost it appears white in color, whereas the soma appears in gray in color. So this is why. Okay, now let us understand this thing, the structure of spinal cord. So in the spinal cord, outside of the spinal cord is surrounded by three meninges. Okay, three meninges will be there. So these three meninges are, they are of connective tissue origin. So meninges are formed from this connective tissue. Okay, and inside this meninges, we have this nervous system, and the spinal cord is made up of many neurons. All these neurons are tightly packed with each other. So this packing of many neurons forming this a spinal cord. And in this thing, what is happening? All the neurons which are present in this spinal cord, all the neurons cell bodies or soma are occupying in this region. That's why this H-shaped region is having gray color. So all the gray colored parts of the neurons, that means all the soma of the neurons are arranged in this H shape. So that this is forming this, all the neurons soma are forming this H shape. Okay, and remaining remaining parts of the neurons, that means axons or dendrites, all those things are occupying the remaining position, a remaining part of the spinal cord. So that's why the remaining part is having this white color. Okay, and coming to brain, in this brain it is vice versa. Outside part is gray in color and inner part is white in color. So all the cells which are present in this brain, almost many hundreds of crores of cells will be present inside our brain. So many hundreds of crores of, okay, many hundreds of crores of neurons are joining together to form this brain. And all those neurons, soma, are outside the brain region. So they are on the peripheral region of the brain. And inside this brain region, all those neurons, axons and dendrites will be arranged inside the cell. So that's why inner part of this brain will appear white in color, whereas the outer part will be gray in color. Whereas in this spinal cord, it is vice versa. Inside will be gray in color, outside will be white in color. So the brain and spinal cord are being formed with the aggregation or with the fusion of many hundred crores of or thousands of crores of neurons. Okay, all those neurons are packing tightly with each other, side by side, one above another. So this packing, this arrangement is forming spinal cord and brain. And how this nerves, whether it is a cephalic nerve or cranial nerve, or whether it could be this spinal nerve. So the spinal nerves are cranial nerves. How they are being formed? Let us see. So we have seen about a single neuron, right? So let us start in this slide from this region. So I think in this slide it is very clear. So let us start from this thing. So this is the axon. Okay. So and the axon is surrounded by myelin sheath. So here you can see that actually this myelin sheath is nothing but a cell. And this cell is surrounding in many layers. So this Schwann cell is surrounding this axon in many layers. See here in this thing, 1, 2, 3, 4, 5 layers are there. So it is wrapping around this axon. Schwann cell is wrapping around this axon in many layers. At the outermost layer, the nucleus will be present at the outermost layer of this Schwann cell. So each one is a Schwann cell. This is one Schwann cell. This is another Schwann cell. So like this, we have studied about the structure of neuron. Okay, so this is a single neuron. Okay, this is a single neuron. Okay, this is a single neuron. So in our nervous system, some neurons are very long that their axons length will be around 1 meter. The axon's length will be around 1 meter. So the you can imagine that. Let us go back to the previous slide. Okay, in this thing. So this is the spinal cord, right? So from the spinal cord, around 31 pairs of nerves are arising, and these 31 pairs of nerves are called as spinal nerves. Okay, each spinal nerve, let us take only a single spinal nerve. See, this spinal nerve originating from this part of this spinal cord. Okay, so this is originating from this part of the spinal cord, and it is extended up to this region. Okay, it is extended up to this region. So you just imagine that, you just remember that the total nerve, okay, the total spinal nerve starting from this region to this region is having a single neuron. The single neuron's axon is starting from this region and it is extending up to this region. So the total length of the nerve, whether it is a spinal nerve or whether it is a cranial nerve, okay, the total nerve is having a long, many long axons will be there. Okay, so throughout the length of the nerve, say for example, in our legs, almost nerves which are present in the legs, last pair of nerves will enter into our legs. Okay, 31 pairs of nerves arise from the spinal cord. Out of them, last pair enters into our legs. So this last pair length will be around 1 meter. Okay, last pair's cranial nerve's length will be around 1 meter. So the axon of the neurons which are present in this last pair will be around same length. So if the nerve is having a 1 meter of length, the axons which are present in the nerve also will be having the same length. So the total originated place of the nerve and ending place of the nerve will be occupying the similar length of the neurons. So in this way, this is a single neuron. This is starting from this originated place of this respective nerve, okay, and it is extending till the end of the nerve. It is extending till the end of the nerve. So in this way, you just remember that if a nerve is there, the total length of the nerve is equal to the length of the axons which are present in this nerve. So in this slide, you can see that this is a single neuron. Okay, the single neuron is covered by here and there. The single neuron's axon, the single axon is covered by this many Schwann cells. These Schwann cells are forming this myelin sheaths. Okay. So the axons are surrounded by this myelinated sheath. So this is a neuron. And what is happening to the soma of this particular neuron? What is happening to the soma of this particular neuron? Let us go back to the previous slides. Okay, see here you can see from the spinal cord, each neuron pair, sorry, each nerve pair, on left side one nerve will come, and right side of the spinal cord another nerve will come. So both these things, they will form a pair of the nerves. So, and again each nerve will be originated due to the fusion of a dorsal root and a ventral root. Dorsal root and ventral root. Okay. So in the dorsal root, all the neurons which are present in this nerve, all the neurons which are present in this nerve, all those neurons, soma or bodies, will be present here in this region. As the soma will be big in size, big in means in terms of diameter, it will be large in size, but axon will be having lot of length. Lengthwise it is big. Okay, lengthwise it is great, but in terms of diameter, this soma are great. So that all the soma are aggregating or accumulating in this region. So, and that's why this is having a some swollen type of structure. So this swollen type of structure is called as ganglia. So remember that in invertebrates also in the nervous system, here and there we will see this ganglion, ventral ganglion. Okay, so in the nervous system of this what cockroach and all those insects also, you have studied about this many ventral ganglia. So ganglia are nothing but, ganglia is the region where all the neuron soma are aggregated. Okay, so here in this ganglion, all soma of the neurons are aggregated. So all the soma are present in this region, and axons are starting from this region and they are extending till the end of this nerve. Okay, so all the soma are pericardia are occupying in this region. They are taking place in this region, and the axons are starting from this region and they are forming this nerve, and the axons are extending till the end of the neuron. Okay, so now let us come again to the how this many neurons are forming this nerve. So nerve is different from neuron. Neuron is a structural unit, and nerve is the a part of we can say that nerve is a organ. Okay, nerve is nerve is an organ. Neuron is a structural unit. So neuron is a unit, structural unit. Nerve is the organ. So we know that all organs, how they will be formed. Cells join together to form tissues. Many tissues join together to form an organ. In the same way, many neurons are joining together to form this nerve. So nerve is different from this neuron. Nerve is different from this neuron. Okay. So in this way, all the neurons, okay, they are covered by myelin sheath, and again all these myelin sheaths, they are covered by a connective tissue called endoneurium. Okay, so again each neuron is covered by endoneurium. In some cases, 2 or 3 neurons may be covered by a single myelin, a single connective tissue. So this connective tissue, in some cases, this may be covering only single neuron, or in some cases it may cover 2 or 3 neurons also, including the supporting neurons. Supporting neurons like glial cells, all those things are there, right? So, including the supporting neurons, the original neurons or important neurons will be wrapped up by a this what connective tissue. So this connective tissue is called as endoneurium. So similarly, many endoneuriums or many neurons, again they will be covered by a another one called perineurium, and this is a fascicle, neuron fascicle. Okay, in muscle cells also, you just remember the structure of muscle cells, skeletal muscle cells, how the structure of muscle cell is there. Similarly, the nerve structure also will be similar to the structure of muscle cell. So this is nerve fascicle. In muscle cells, in muscle structure, we call it as muscle fascicle. Here it is nerve fascicle. So in nerve fascicle, many neurons will be there. The total fascicle will be covered by a perineurium. And in the perineurium, many neurons will be there. All these neurons, again, they will be covered by another wrap-ups. They are called as endoneuriums. So like this, again, these many perineuriums, again, they will be covered by another finally last layer of connective tissue called as epineurium. So at the outside of this now, epineurium is there. Inside epineurium, many bundles will be there, fascicle bundles. Each fascicle bundle will be covered by perineurium. And again, in each bundle, many neurons will be there. All these neurons will be covered by endoneurium. So, like this endoneurium, perineurium, and epineurium are there. And again, these neurons, these nerves will be supplied with circulatory system, blood circulatory system. So, this what arteries will come and they will enter inside this thing, and in between these fascicles, this they will travel with this along with these fascicles. While going along with this travels, while traveling along with these fascicles, this artery, this red color one artery, it further bifurcates into many this what meshwork like structures, are what we call them capillaries. All those capillaries, they will supply nutrients to this thing, and again in return it will collect waste materials like urea and carbon dioxide and all those things, they will enter into this venule, and again all these venules, they will come out of this thing in the form of a vein. So arteries and veins will be supplied into this fascicles, supplied into this neuron. Similarly, brain region also will be supplied with this thing. We have studied about those connection with nervous system and blood circulatory system in last year. Okay, so in this way, this is the arrangement of neurons. So how these neurons are arranged to form these nerves. So I think now you are familiar with this arrangement of this structure of this nervous tissue. Nervous tissue. So this is the nervous tissue present inside our body. It is forming three parts: CNS, PNS, and ANS. Okay, and CNS, PNS, all these things are, and ANS, all these things are forming with the fusion or arrangement of these neurons only. Okay, so the neuron nerves. So these are the nerves. Parts of this PNS are called as nerves, cranial nerves and spinal nerves. How these are formed, we have seen in these two slides. Okay. So again, coming to this thing, this is very, very important to understand, remember, to better understand the next class, that inside this now and inside this now, what is there? Fluid will be there. Okay. All this is nothing but this is a tissue, nervous tissue. Okay. So this nervous tissue is supplied with arteries and veins. So arteries and veins, outside this arteries and veins, what will be there? Outside this arteries and veins and surrounding this neuron, surrounding these fascicles, what will be there? Extracellular fluid will be there, or tissue fluid will be there. Tissue fluid or extracellular fluid will be there. So all these neurons, all these fascicles are surrounded by a fluid called extracellular fluid. Okay, so inside this neuron, cytoplasm is there. Outside these neurons, okay, outside these neurons, even though they are surrounded by peripheral, endoneurium, even though they are surrounded by endoneurium, they will be having connections with this what? Extracellular fluid. So this is very, very important to understand next class, that each nervous system inside this each neuron, inside the neuron, cytoplasm or neuroplasm will be there. Outside the neuron, extracellular fluid will be there. So this is very, very important, you remember this thing. So with this, today's topic is over. I think now you better now you have understood very well the arrangement and structure of this nervous system. Anyway, this is not a part of our syllabus, but it is very, very important to understand this thing. Okay, so in our syllabus only we have this thing, what structure of neuron and types of neurons. Okay, in types of neurons we have bipolar, unipolar, multipolar neurons, and depending on in which direction it is sending signals, sensory neurons, motor neurons, mixed neurons, and interneurons. And another type of classification based on presence or absence of myelinsis, depending on this thing, myelinated, unmyelinated. So this slide is part of our syllabus. This slide is also part of our syllabus, and this one, these are part of our syllabus. All the remaining things are not important in terms of our syllabus or in terms of examination, but they are very, very important for us to understand the next topic. Okay. So with this, today's topic over. Next, we'll meet with another topic in next class.